In the light-dependent reaction, light strikes photosystem 2, which causes electrons to become excited. The electrons leave photosystem 2 and continue down the electron transport chain. The photolysis of water occurs when it is split to replace the electrons lost when photosystem 2 is struck by light. The product of the breakdown of water is hydrogen ions and oxygen. The electrons continue down the chain, and their energy is used to pump hydrogen ions, otherwise known as protons, from the stroma into the thylakoid space. The result is a proton gradient because the stroma has a low concentration of the hydrogen and the thylakoid space ultimately has a high concentration. The hydrogens, in order to move back from the thylakoid space back to the stroma, they have to go through ATP synthase, which is a type of protein. When the hydrogen ions move through the ATP synthase, it rotates, which makes ATP, which is used in the Calvin cycle. So as the electrons move from the protein pump, they lose their energy. So at photosystem 1, it's struck by light again, which excites them. The electrons then move to ferredoxin, which is a protein where NADP is reduced to NADPH. And this transformation from NADP plus to NADPH, reduction means that the NADP is gaining a hydrogen. So in summary, the light-dependent reaction uses water to make oxygen and also ATP and NADPH, which are used in the Calvin cycle, to make organic compounds. And then the Calvin cycle starts with when Rubisco, an enzyme, adds a CO2 to an RUBP, making it a 6-carbon molecule. And then immediately it is split into 2 3-carbon molecules called thioester 3-phosphates. And then in the stroma, the 2 3-GPs, or G3Ps, the glycerate 3-phosphates, they are reduced to 2 triose-phosphates, which happens when 2 ATPs are reduced to 2 ADPs and 2 NADPHs are reduced to 2 NADPs. These 2 NADPs are used in the light-dependent reactions. So then when forming glucose, 1 carbon is removed from the triose-phosphate, like the pairing. So only 1 out of the total of 6 carbons, but glucose has 6 carbons total. So only 1 is added at a time. So then there are 5 out of the 6 original remaining carbons, and using ATP, which then gets changed to ADP, the 5 out of 6 carbons, so the triose-phosphates, are regenerated into RUBP, which is the 5-carbon molecule, which is used in the beginning of the cycle. So to make a full glucose molecule, the whole cycle has to repeat 6 times, so that there are 6 carbons. And that is the end of the Calvin cycle.